Our next presenter today is Amber Johnston. She's a second-year master's student with the Michigan State University Aquatic Animal Health Lab, the Aquatic Animal Disease Ecology Program. She's interested in the intersection between fish health research and fisheries management, specifically how fisheries managers and fish health researchers can collaborate to reach a common goal of healthy fisheries. So thank you for joining us today, Amber, and you can take it away.
Yep, I thought my cats were gonna calm down for this, but they're obviously not going to, so I'm gonna do my best under the circumstances. So I'd first like to start out by acknowledging my co-authors for their contribution on this research that I'm extremely excited to share with you all today. And I also want to take a minute, I'm so sorry, I also want to take a minute to thank the organizers of these virtual seminars, because now as a second-year master's student, I haven't had many opportunities to present my work in person. So without these virtual opportunities, I really wouldn't have had a way to get my work out to the greater fish health community. So to kick things off, lake sturgeon are the only native sturgeon species in the Great Lakes, where they're an ecologically, culturally, and historically important fish. And unfortunately, since the 1800s, when they became a valuable commercial fishery, their numbers began dramatic declines, and now lake sturgeon populations are estimated to be less than 1% of historical numbers. And because of this, substantial resources have been allocated to the hatchery rearing of lake sturgeon for conservation purposes. So what exactly does that look like? Well, hatchery rearing of lake sturgeon for restocking into the Great Lakes is now a common practice around the basin. And some of these facilities, pictured here, are populated exclusively from gametes collected from wild adults. And unfortunately, these facilities are not exempt to troubles of their own. Whether they're state, federal, or tribally managed, many of these facilities grapple with occasional mortality events, some of which near 100% cumulative mortality. And one of the unique things about lake sturgeon, especially in the context of the Great Lakes, is how prehistoric they are. And from what we know of other sturgeon species, many of the pathogens that affect them are quite distinct from other fish. And compounding that, many can't be detected without specialized and specific diagnostic tests. And possibly because of this, when we see these mortality events, we look at the cause by using our routinely employed diagnostic techniques. But because some of these pathogens can't get picked up, many of these mortality events go unexplained. And while this is happening, other sturgeon pathogenic microbes seem to be increasing in host and geographic range. And despite an apparent encroachment towards the Great Lakes Basin, the potential impact of these sturgeon pathogenic microbes, should they end up here, is largely unknown. And unfortunately, because we can't pick them up with our routinely employed diagnostic techniques, we know very little about their potential presence in the Great Lakes Basin. And that's where my thesis research is hoping to uncover some unknowns. My ongoing thesis research is a part of a larger project that looks to assess the risk of emergent and endemic fish pathogens to Great Lakes lake sturgeon populations. So for the next few slides, I'm going to bring you up to speed on what's been done as a part of this larger project before diving into some of my most recent findings. And some of this might look like review if you attended my talk last year. So to begin addressing these unknowns, I set out in 2019 to sample adult lake sturgeon from the St. Clair River and the Lake Erie watershed and the Black River and the Lake Huron watershed. And during year one, I non-lethally sampled an array of tissues and fluids from over 200 adult lake sturgeon. And we collected these samples with the intent to employ specialized diagnostic methods aimed at the detection of a wide array of microbes, including sturgeon pathogenic microbes. During year one, we observed these circular, raised, semi-translucent areas of apparent cutaneous thickening, and these lesions varied in location and ranged from focal to multifocal to coalescing in nature. Additionally, the lesions varied in severity of skin coverage, whether it was a small area on a fin or widely dispersed along the body of the fish. And in terms of how frequently we saw these lesions, they were observed at a prevalence of 5.8% in the Black River and at a prevalence of 1.3% in the St. Clair River. Both are in different Great Lakes watersheds. So upon doing some digging into what could possibly be causing these lesions, I came across multiple references referring to acetopencered herpes viruses 1 and 2, both in the family Alloherpes viridae. And based on the literature, the lesions I'd collected from adult lake sturgeon appeared grossly similar to those noted in other sturgeon species. And for that reason, I began pursuing specialized diagnostic techniques centered around the detection of acid-censored herpes viruses. So armed with that information, I optimized a PCR using a primer set designed by Dr. Larry Hansen and colleagues that targets the DNA polymerase gene of a range of fish viruses, including those in the family Alloherpes viridae. I screened these lesions using this PCR and successfully detected an amplicon from an unknown virus associated with these lesions. I then sequenced the amplicons and analyzed them phylogenetically, and what I'm showing you here is the resultant dendrogram. Highlighted on the right are my sequences, and from this, we determined that we definitely found an alloherpes virus, and it was most similar to, yet still distinct, acid-censored herpes virus 1. And while I found genetic traces of a virus, unfortunately, and despite my best efforts, I was unable to isolate this virus in vitro, which is critically important for further studies. And though disappointing, it wasn't particularly surprising given that viruses within the family Alloherpes viridae are notorious for being difficult to isolate via cell culture. So why exactly was this finding so significant? Acid-censored herpes viruses 1 and 2, also known as white sturgeon herpes viruses, are listed as emergency pathogens on the Great Lakes Fishery Commission's Fish Health Model Program. And they earn their spot on this list due to their ability to cause mortality in other sturgeon species and their potential to be transmitted from adults to offspring. So shown by its listing in the model program, there are currently no published reports of an acid-censored herpes virus in the Great Lakes, yet now it seems as though we've detected a presumptive acid-censored herpes virus. And while this research began to answer some critical questions, it raised many, many more. What exactly was this acid-censored herpes virus? We couldn't isolate it in 2019. How can we modify and isolate it next time we sample fish? Could it be contributing to these long-standing population declines? And could it be behind these undetermined mortality events in the breeding facilities? And that brings me to my overarching study hypothesis, which is that a Great Lakes acid-censored herpes virus is negatively affecting the health and survival of Great Lakes lake sturgeon. So to answer our hypothesis, we developed 3 objectives that I'll go through one at a time. The first objective is centered around elucidating the effects this acid-censored herpes virus could be having on wild adult lake sturgeon. So to begin understanding the effects of this virus, we went back to look at tissues collected from those lesions. And you may remember some of these images from last year while I was early in the analyses, but I'd just like to go through them one more time. So I'd seen gross changes in the skin caused by these lesions, but I wanted to know what was happening microscopically. But before we get to exactly what I found, here I'm showing you a skin section collected from a grossly normal area in which you can see an epidermal layer of normal thickness and the underlying dermis. Now what I'm showing you here at the same magnification is tissue directly from a plaque-like lesion, and we immediately noticed how thickened the epidermis was, and we also noted inflammation in the epidermis and the underlying dermis. Additionally, we found evidence of tissue damage in the form of swollen, degenerate epithelial cells noted with the arrows. And we also found evidence of intracellular edema or fluid accumulation between the epithelial cells. And collectively, what this told us was that in lesions where we'd found the virus molecularly, we had evidence that there was tissue damage there was damage at the tissue level, specifically in the skin. And this is particularly important given the role skin plays in pathogen defense and host osmoregulation. So because of this, we concluded that this virus could possibly be having negative effects on wild adult lake sturgeon. So our second objective was to isolate the virus in vitro. And while the detection of genetic material was promising, one of the pillars for linking a virus to disease causation is the fulfilling of Rivers' postulates, which requires infectious virus experiments under controlled laboratory conditions, something that's substantially easier to do when you can cultivate the virus. So now that I knew that I was dealing with an alloherpes virus, I was able to research different methods to effectively isolate viruses in this family. And I was super excited to get out into the field in 2020, though the ongoing COVID-19 pandemic certainly posed challenges. Despite that, though, through perseverance and collaboration, I was able to get out to the Black River and non-lethally sample 79 spawning adult lake sturgeon. I just want to say that hiking a river decked out in the proper PPE certainly wasn't the ideal field experience, but it's something I'm thankful for nonetheless. So moving forward to what I saw while I was in the field, I saw multiple fish again exhibiting these acid-pentoid herpes virus-linked lesions in varying severities like the year prior. And these lesions were observed at about a 15% prevalence. So now that I had these lesions and I'd done research to develop new sample handling techniques, I was finally able to employ my new virus isolation methods. And based on my research, I was very optimistic that they'd work. So after implementing these modified protocols, I inoculated lesion tissues onto several cell lines derived from multiple fish species, including sturgeon. And soon after, I saw evidence of a replicating agent in the form of multinucleated giant cells, or syncytia, noted here with the blue arrows. Eventually, that became even more widespread, as you can see here. And this finding was very encouraging because last year I didn't get this far, and I was seeing that my modified protocols worked this year. So since we saw evidence for a replicating agent on cells, we wanted to take a look at them via transmission electron microscopy. So when looking closer at the affected cultured cells, we appreciated enveloped icosahedral-shaped virions, which in 2D appear as hexagons and also resemble other alloherpes viruses. Excitingly, when we looked at the tissues themselves, we saw the same icosahedral-shaped virions, though in this photo, it's much easier to appreciate the surrounding envelope and electron-dense core. And this suggested that the replicating agent in the cells was likely the same virus as that in the skin lesions, at least ultrastructurally. And this was a very exciting finding. So to further confirm what we had in the cells, we assayed the affected cells using the same PCR assay as the year prior that targeted the DNA polymerase gene, and once again got similar positive results. So backpedaling a little bit to what's done with those amplicons before sequencing, when using a PCR assay that amplifies multiple targets, there's always a risk of non-specificity. And cloning lets us take that into account and be more confident with what we're submitting for sequencing. So for those less familiar with the cloning process, it begins with an unpurified PCR product that's then purified, inserted into a cloning vector, plated onto selective growth media, and the resulting growth consists of clones of the PCR product inserted into the vector. And again, this all just lets me be more certain that what I generated here is my target and not in fact something else. So what I'm showing you here is a table that's going to be depicting the origin of the generated sequences, whether they came from tissue culture, skin lesion, whether they were cloned or not, what year the samples were collected, and the sample's percent similarity to the acid-pentoid herpes virus 1 DNA polymerase gene. So in 2019, what I saw was that all of my sequences sampled resulted in 79 to 81% similarity to this acid-pentoid herpes virus 1 DNA polymerase gene. Like I mentioned previously, I was not successful in isolating the virus, so I don't have any tissue culture samples from 2019. But in 2020, I pursued the same analyses, though I had been able to isolate the virus. So what we see here excitingly is that whether the samples were from the lesion itself, tissue culture supernatant or cells, cloned skin lesions, or cloned tissue culture samples, all of these samples across both years exhibited a similar 79 to 82% similarity to the acid-pentoid herpes virus 1 DNA polymerase gene. And not only were these sequences similar in their similarity to acid-pentoid herpes virus 1 DNA polymerase gene, they were also similar amongst each other. So to give a better picture of this, here's the resulting dendrogram from all of those sequences. I'd first like to point out my reference strains that I used, some of which I downloaded from GenBank, others that I'd gotten sent to me from collaborators at UC Davis and sequenced under our laboratory conditions. And those were acid-pentoid herpes virus 1 and 2 sequences. And here's what's exciting: whether these sequences were generated from tissues, tissue culture, or cloned to the like from both years, they all fell within this large, robustly supported clade. And since these sequences were still robustly supported as distinct from acid-censored herpes virus 1, we concluded that we could be dealing with a potentially novel Great Lakes endemic acid-censored herpes virus. So while we were beginning to address some of the unknowns about this virus, a critical question was still left unanswered. What effect could this potentially novel virus be having on the survival of juvenile lake sturgeon? Could it be causing problems in these previously mentioned rearing facility mortality events? I had a cat just meow at me, sorry. So to begin addressing those questions, this year and last, I collaborated with the Black River Sturgeon Rearing Facility to rear juvenile lake sturgeon and held them under quarantine conditions at the University Research Containment Facility here at MSU. And now that we also isolated this virus and could readily grow it, the possibilities felt endless compared to where I was in 2019. Though based on our phylogenetic analyses, we were dealing with a potentially novel virus in a new host. And with so much unknown, we set out to evaluate the effects of dose, temperature, and exposure, all in an effort to guide full-scale challenge models. So with those variables in mind, I organized a series of small-scale pilot challenges. I evaluated the effect of dose by looking at a high and low dose via IC injection. I evaluated the effects of exposure route by doing a high and low dose immersion challenge. And I evaluated the effects of temperature by doing a 22-degree IC injection challenge compared to the other 10 to 11-degree challenges. And while done unreplicated, all treatments had a corresponding negative control. After exposure to the virus, I monitored fish for the development of disease signs. Over time, I began to notice a series of things. First, I noticed perioral hemorrhaging, noted here, and varying degrees of exophthalmia, both of which were noteworthy because some of these signs have been reported in sturgeon infected with other acid-censored herpes viruses. We also observed skin lesions that were ulcerative in nature in infected fish, and these skin lesions were observed at varying intensities. We found some that were more focal in nature and other that were slightly larger or multifocal to coalescing. And excitingly, we did re-isolate virus from both lesions pictured and confirm the presence via PCR. So now, based on these pilot studies, I saw that the virus could induce clinical signs of disease, but what effects did it have on survival? What I'm showing you now is a cumulative mortality curve of all pilot challenges conducted. On the y-axis, I have cumulative % mortality, and on the x-axis, I have days post-infection. And one thing I'd like to draw your attention to in particular is this high dose, low temperature injection challenges. In this challenge, there were no mortalities for the first 50 days of this study. And then suddenly, within 4 days, cumulative mortality went from 0 to 60%. And a few weeks later, from 60 to 80%. There were also no mortalities in the negative control. And from this, we gathered that in these conditions, the course of disease could be slow moving with this virus. And I'd also like to highlight that in most instances where a group of fish was exposed to virus, cumulative mortality was at least 20%. So before I move on, I have one more recent finding regarding my pilot challenges to share. In the previously mentioned high-dose, low-temp injection challenge group, where percent mortality reached 80%, there was one remaining survivor. And these pilot challenges were recently ended, as pilot challenges go, and fish examined, and that particular treatment group remained exciting. Upon examining the sole survivor of that group, over 300 days post-infection, I noticed this opaque plaque-like lesion. And while tissue culture and histological analyses are still underway, the similarity in gross appearance between this lesion and those I'd observed in adults was uncanny. Overall, the results of these pilots were very telling and concluded that this virus can induce mortality in juvenile lake sturgeon. Additionally, they're helping me guide full experimental challenges, some of which are currently underway or on the docket to begin. So to wrap things up, we have a few key conclusions. First is the first report of an acid-censored herpes virus in Great Lakes lake sturgeon in Michigan. As I said before, there are currently no published reports of an acid-censored herpes virus in the Great Lakes, and given the devastation viruses in this family have caused other sturgeon populations, as well as their status as emergency pathogens on the GLFC Fish Health Model Program, this finding is very significant. Second is the first in vitro isolation of an aspen-centered herpes virus in the Great Lakes. And while initial attempts at isolation were unsuccessful, using sturgeon-specific cell lines and modified protocols, our research crew is now able to successfully isolate, cryopreserve, and revive virus for further studies. And third, through that work, I've begun to elucidate the effects this virus has in adults and juveniles. And I've found that this potentially novel virus in the adults, it's causing skin damage. And I've shown that this virus in juveniles presents similarly in the skin. And unfortunately for lake sturgeon conservation, this virus can induce mortality in juveniles. So that begs the questions, has this virus been working in the background, contributing to these declines in lake sturgeon populations? Or has it been a factor in these unexplained mortality events? My research now is positioning us to answer those critical questions. So with that, I'd like to thank the U.S. Fish and Wildlife Service Great Lakes Fish and Wildlife Restoration Act for funding my study, the Black River Sturgeon Rearing Facility crew, Michigan DNR, MSU Aquatic Animal Health Lab members, and my major advisor, Dr. Thomas Locke. So thank you all for listening and getting to see my cat for the last 20 minutes.
Excellent presentation, Amber. It's a very, very impressive project working on a difficult virus. And hello to your cat too.
Yeah, I was hoping they'd be better.
So folks, feel free to unmute yourself or drop a question in the chat box. I've got one to kick things off for you. Can you give us a little more background on the sturgeon management, how the sturgeon, if these watersheds participate in the propagation programs and if fish are being moved around, and could there be a link that way?
Yeah, so I'm not incredibly familiar with the exact like sturgeon management, like what's all going on in the Great Lakes, but I do know that a lot of the facilities, the fish that are collected from a certain stream system, they're reared and restocked into that system typically. I know it varies from site to site, but in general, all the sturgeon work that's being done is strictly for conservation purposes. So every fish that's reared is released back into the Great Lakes Basin.
Okay.
I can't say exactly if like where the gametes come from, go exactly where the juveniles go. I don't know that for sure. I know that's the case in a lot of situations, but I don't want to say every. So
yeah, I suspect that'll be a question coming up as a new virus shows up in two different places at the same time.
Yeah, exactly. And it's hard because all these watersheds are connected. So you have all of these different Great Lakes in different places working with them. I mean, I found it in 2 different watersheds, but that's not to say it's not in other ones as well already. So.
All right, so we have some questions here in the chat from Steve Backman. Do you plan to do any post-recovery challenges?
So post-recovery is in what virus I recover, reinfecting fish with that. That's currently not anything that we have on the docket right now, but that's definitely interesting. So I'm not sure if that's what he was talking about. If not, please clarify and I can see if I can better answer.
Yeah, feel free to clarify, Steve. He may mean if there's surviving fish that have been challenged, if you re-challenge them.
So that's not currently something we're looking at right now. We have thought about potentially stressing the fish to see if that would change, if we're able to recover virus or not from them. Okay, yeah, so I get that now, but that's not currently something we're looking at doing, but it's not out of the question. I mean, this is a potentially new virus that we're dealing with. So there's a lot that we want to do. We just have to figure out what we have the means to do and time to do.
Yeah. We have a question here from Dave Groman. He's, all right, so has this herpes virus been isolated from fish at the hatchery?
I've only isolated it so far from wild adult lake sturgeon.
Okay. And then his follow-up question is if you've looked for other disease conditions such as iridovirus in the lesions.
Yep. So currently we've only looked for these aspen-centered herpes viruses. That was the first key point we want to tackle. Other things in the study that I'm going to be working on in this coming fall is going to be looking for things like white sturgeon iridovirus and some of those nucleocytoplasmic large DNA viruses. So there is more to come, but right now we're currently working with the CHVs.
Great. Okay, we got another question here in the chat. What were your results after exposing the sturgeon to different temperatures?
So we saw mortality quicker in one of the virus infected tanks, but in terms, again, these were done at really small scale. So I didn't recover virus from the virus infected fish that had been a mortality. So I didn't see much of a change, but it could also have to do with the age of the fish at which they were challenged. I had done that first cool temp challenge where I had that 80% mortality with fish that were about 10.5 weeks old. Whereas that high temp challenge, I wasn't able to get it underway until the fish were 24 weeks old. So as a lot of us probably know, herpes viruses tend to do the most damage in young fish. And as the fish get older, they don't seem to be as affected. So there's a lot of stuff at play, and it also has to do with the fact that growing this virus isn't the easiest thing in the world. So sometimes we're at bay of when we're able to start things and when we're able to end things. So while we didn't see any significant, I say significant loosely because with those unreplicated pilots, it's hard to use significance in any place, but we can't say it was because of the age or because of the temperature or what. So
now, follow-up question on the conditions there. How the experimental conditions that you tried, how well did they match with the hatchery system? Were those temperatures
that was
the same? Or?
Yeah. So that was the big reason we wanted to do the high temp challenge, is because the higher temp more closely aligned with what they'd be seeing in the rearing facilities. So it's something that we haven't like thrown out yet. It's still like on our radar as something we'd want to try with the full scale challenge potentially. But the ambient water temp at our facility is that 10 to 11 degrees Celsius. So. Okay.
Any other questions out there? Feel free to unmute yourself or put them in the chat.
Yeah, Amber, this is John Drennan from Colorado Parks and Wildlife. A great talk. And Dave actually asked one of the questions about whether you've seen this in the hatchery or not, and it appears that you haven't. And then the other question that I was going to ask you was about your life stage of challenging the virus, which you did it at a certain age. Can you correlate anything with recruitment of, I'm sorry, did I say white sturgeon, lake sturgeon in those places with declining numbers and/or, so you did a challenge at a certain age, and it was high dose. If you did an earlier life stage challenge with a lower dose, would you see more morbidity and mortality? It looks like just what you said, as the fish gets older, it gets more refractory to mortality, even though you can see some lesions. But does that correlate to what you see out in the field with recruitment of age classes?
I unfortunately am not very familiar with a lot of the recruitment work that's getting done right now. I know there's a lot of it getting done at the Black River Rearing Facility here in Michigan, but I haven't been exposed to that much. So, but you're completely correct in that this virus affects fish of different ages in different ways. So we're hopefully going to be seeing how that could change with younger fish versus the fish I currently have infected. So I'm sorry I can't answer your question as they relate to one another.
No, anyway, it was a great talk. Yeah.
Thank you so much.
And John, I'll just jump in for a second. And I think I did see that Dr. Kim Scribner was on the line, but I think there's a lot unknown right now in terms of where potential bottlenecks in recruitment would be. But generally, that those very early life stages, I think, are where people are most concerned with larval drift. And so, yeah, I mean, at least, Amber's working with the virus that we know very little about, but at least what we know, based on some of the work you did too, John, with, ACHV1 and 2, it does seem like you can find the virus in the reproductive fluids, and it does seem like it can do quite a bit of damage at early life stages. So I think there is risk. We have no idea. But if you operate based on what you know for other herpes viruses, I think, we're most concerned about those early life stages.
Yeah, thanks, Tom.
All right, any other questions out there? Okay, well, thank you very much, Amber. That was an excellent presentation. Virtual round of applause to you as well.
Thank you so much.